the structure of proteins. In this video, we're going to talk about proteins. We're going to see how a body makes a protein. And in the process of making a protein, what are the levels of protein structure? We're going to introduce the protein data bank. And we're going to talk about how we can quantify the primary structure of a protein using graph theory. Proteins are large molecules. Sometimes they're called macromolecules. And they are also sometimes called the workhorses of the cell. When we think of biological functions, the key word here being function, it is these protein molecules that carry out these functions. For example, a function might be to transport ions across the cell's membrane. Then there are proteins this is their function. They have a channel. Some of them even act like a pump. And there are other proteins that transport different proteins around the cell. So you can really think of a cell as a bustling city. And all of the city workers are the proteins. So how does the body make a protein? We have probably all heard about our genes. And a gene typically encodes a protein. What does that mean? It means the DNA sequence is a set of instructions for making the protein. How is that possible? Well, first, when the gene is activated, what happens is a copy of one of the strands of DNA is made. This is called transcription, and the copy is called RNA. Notice that RNA is not identical to DNA because the letter C, if I look at the top line of the DNA, the first three letters are G, T, G. But in the RNA, we have G, U, G. So the letter T is replaced with U. If we look at the next block of three, we have C, A, T. So again, the letter T is replaced with U. The other letters are the same. This is also notice that we are reading these in blocks of three. So each block of three is then translated to represent a unique amino acid. So GUG translates to V. And CAU translates to H, where the V and the H and the L on the very bottom is a sequence of unique amino acids, which we'll talk about. There are three levels of protein structure: the primary, the secondary, and the tertiary. The primary structure is the sequence of amino acids. That's what we saw on the preceding slide. Following the transcription and then the translation of the RNA sequence, we have a sequence of amino acids, and that sequence is called the primary structure. So, for example, below we have a sequence beginning with the letter M, and M, D, F, R, etc. So that is the primary structure of a protein molecule. Now, what does an amino acid look like? Recall the primary structure is just a sequence of amino acids. The structure of an individual amino acid consists primarily of two parts: the backbone and the residue. The backbone is shown on the left and is everything except the block labeled R. All amino acids have the same backbone structure. And then the part labeled R, which is the residue, is different for each of the 20 amino acids. These, the backbone structure, are able to link to each other. So you can think of it like a charm bracelet. You have the chain, and then you have the charms hanging off the chain. And residues, then, are the charms. Here we have a schematic drawing of each of the 20 different residue structures. I'm pointing to, on the right, the central C, that is the central carbon atom that is in each of the backbone structures. And then the residue starts where I'm pointing with the capital R. So the central carbon is sometimes called the C alpha, and then the carbon atom immediately below is the first carbon of the residue chain, or the residue structure. Notice here we have like an angle. Each angle represents a carbon atom. So when we represent an amino acid as a graph, where each vertex is an atom, we would actually have a vertex at that angle. Here we can see the residue structure of each of the 20 amino acids without the backbone. So we just start the drawing with the very top. We understand up there is a carbon atom, which is the central carbon, and we have labeled each of the angles with the letter C to represent the carbon atom. Notice that as you look towards the bottom of the residues, then we have atoms other than carbon. So for example, with amino acid Q, we have an oxygen and a nitrogen atom. As I said, we can represent each of these as small graphs, and we can label each vertex by its corresponding atom. Now, moving from the primary structure, what happens is if we have this sequence of amino acids, then physically we have a chain of amino acid structures. So this depiction shows that each of these circles is representing the amino acid. As this amino acid structure is produced in the cell, one way it's described is that the residue structures repel or attract each other. And so you get this movement where some are pushing away, some are sticking to each other, and a different structure starts to be formed, and these are called secondary structures. Some cases you get a twisting, which is called an alpha helix. And in some cases, you get a sheet that buckles, which is called a beta strand. And in some cases, you just still have the chain connecting these, which are just called loops. So the alpha helices, which is the twisting, and the beta strands, all come together for this three-dimensional molecule. And that is called the 3D structure. So in the picture on the right, we can see the secondary structures. This is sometimes called a cartoon representation because it makes it very clear to see the alpha helices and the beta strands and the loops, and those are all the secondary pieces, but the entire molecule is the tertiary structure. With the completion of the Human Genome Project, we learned that there are more than 22,000 genes in our genome. And since each gene encodes a protein, we know that there are more than 22,000 proteins. However, the structure, and therefore the function, of these proteins is not known. For some of them it is, but not for all 22,000. So, given the primary structure of a protein, can we determine its tertiary structure and therefore have some insight into its function? And this is a big area of research in computational biology. The Protein Data Bank does contain information on the proteins that we have determined the 3D structure of. So if in the laboratory we have the three-dimensional structure determined by technology such as X-ray crystallography, then once that is determined, the 3D coordinates of every single atom in that protein structure is recorded and deposited in the Protein Data Bank. So we've talked about proteins, protein structure, what proteins do, and that proteins are the functioning units within each cell. We have discovered that the three-dimensional structure of all proteins is not known, but those that we know are deposited in what is called the Protein Data Bank. In the next video, we're going to talk about the Protein Data Bank.